Welcome to the Secret Science Club. I'm Dorian Devens.
I am Margaret Middlebach.
And for those of you who are Secret Science Club regulars, welcome back. For those of you who are unfamiliar with us, the Secret Science Club is a public lecture arts and performance series featuring scientists who study everything from black holes to the human brain. Our live events have been going on for 16 years in New York City. We're now online with expanded audience reach and more frequent events. Here are a few logistics. Please keep your video and microphone off during the presentation. Our speaker, Salvador Almagro-Moreno, will be answering questions after the presentation. Our cocktail for the evening is the Micronaut, designed by the Secret Science Club Experimental Mixology Lab. The recipe can be found on chat and also on our website. If you're not a member of the Secret Science Club yet, you and your friends can sign up on our website at secretscienceclub.blogspot.com or by sending us an email to secretscienceclub@gmail.com. If you're on our list, you'll be the first to know of upcoming events. This, by the way, is a free event, as you know, but we're a nonprofit and we can really use your support. You can donate to the Secret Science Club via donor box using PayPal, credit card, Apple Pay, and Google Pay. The donor box link is at our website at secretscienceclub.blogspot.com and in the chat here. As always, we want to thank everybody who supported the Secret Science Club throughout this year and the millennia. So now I turn it over to Margaret, and she's going to introduce our speaker for tonight.
Hello, everybody. Thank you, Dorian, and thank you, everyone, for coming to Secret Science Club online tonight. We are very pleased to have Salvador Almagro-Moreno here with us as our speaker. Dr. Moreno is a molecular biologist and an assistant professor of medicine at the Burnett School of Biomedical Sciences at the University of Central Florida. His research focuses on the emergence and evolution of bacterial pathogens that can severely impact human health and human populations, a topic that has taken on renewed urgency in light of the current COVID-19 pandemic. His lab is both innovative and multidisciplinary, combining insights from molecular biology, genomics, phylogenetics, research on pathogenesis, and ecology. He's the recipient of numerous awards and honors, including the National Science Foundation Career Award, the Burroughs Wellcome Fund's prestigious Path Award, as well as recently being named a Scialog Fellow. This month, Dr. Moreno was the author of the cover story in American Scientist magazine. His article addresses the question, can scientists predict where disease-causing microbes will arise before they cause the next pandemic? And that's part of what he'll be talking about tonight, as well as discussing the work of his own lab. We're very happy to welcome Dr. Salvador Almagro-Moreno to the virtual stage here at the Secret Science Club. Please take it away, Dr. Moreno.
Thank you so much for the kind introduction, and thank you so much for the invitation. Cholera, the poverty drink, and holy water of those that only get baptized when the levees break. In the cradle of hunger, some children are death-fed. Cholera, mayhem of the matter. In the cradle of hunger, some children are death-fed just by drinking water, just by drinking water. I wrote this in one of my visits to the International Center for Diarrheal Disease Research in Dhaka, Bangladesh, where I have a collaboration studying cholera dynamics. What's cholera? Cholera is a severe diarrheal disease. It's transmitted by drinking fecally contaminated water or food. Here you have a postcard from the 19th century showing what's called, they called King Cholera, providing water, contaminated water to people in London from the Broad Street Pump, which if you have not read the book, The Ghost Map, I highly recommend it because it's like how the field of epidemiology and urban planning got started. And cholera causes death due to extreme dehydration and electrolyte loss. And just a simple math can illustrate how severe this is. If you are, let's say, 75 kilograms, and remember, one kilogram 1 liter of water is about 1 kilogram. So like 25 kilograms of your body is going to be like bone mass, right? So you have like 50 kilograms of flesh. Imagine losing 20 of them within a day. So that's how devastating the disease is. Here you have on this picture what's called the cholera cot. So basically, it's a pretty straightforward system where you have a bucket that collects the feces, and you have a place to provide intravenous fluids and also provide oral rehydration salts. Treatment is really effective. So it's almost like the mortality rate drops almost to zero once the patients can get the treatment. However, the disease remains endemic in many nations that have limited access to clean drinking water and proper sanitation practices. There are approximately 3 million reported cases of cholera per year in endemic areas, and it causes about like $100,000 reported deaths. This is severely underreported, as you can imagine, because of many reasons, from inadequate reporting to fear of losing trade and tourism. What makes this even more tragic is that the greatest burden occurs in children that are younger than 5 years. So cholera is one of those few pandemics that we've had, caused by a small select group of organisms. There's been so far 7 pandemics of cholera. The first 5 occurred within the 19th century, and the 6th and the 7th, which is the one that it's ongoing, happened between the 20th and nowadays in the 21st century. The disease is caused by a bacterium called Vibrio cholerae 01, which is as you can see by its shape, it's a motile and comma shape. That Vibrio actually stands for comma, and it's a natural inhabitant of aquatic ecosystems. What's really interesting about this pandemic disease is that only a handful of strains from that species can cause cholera in humans, can cause pandemic cholera. What you're looking at here, it's a phylogenetic tree. Basically, it shows the evolutionary relationships of different strains of cholera, of Vibrio cholerae. On top in blue, you have like some environmental strains that were isolated from estuaries, ponds, so forth, that cannot cause disease in humans. And at the bottom, in red, you have this pandemic group. As I said, only a small number of them are capable of causing disease in humans, but all of them emerged and evolved from an environmental ancestor. That is not a rare thing. In fact, the vast majority of these pandemic diseases emerge from an harmless ancestor that lived in the environment, didn't cause any harm to humans, and evolved to manage to not only colonize us but spread worldwide. One of the most famous one, it's the plague. The plague is caused by a bacterium called Yersinia pestis. And this one is transmitted by fleas, in the case of the bubonic plague, or from person to person, in case of the pneumonic plague. And it has a brutally high mortality rate. We're talking about almost 70%. And it really depends on the susceptibility of the patient, but it can reach up to 90% mortality rate. And I'm sure you've seen this picture of the plague doctors wearing these masks, thinking that they could be protected from the disease. The disease truly devastated the Eurasian continent in the 14th century. In only 7 years, between 1346 and 1353, it killed between 75 and 200 million people. On today's numbers is really high, but think about 6-7 centuries ago. We're talking about between 30 to 60% of the European population and 35% of the population of the Middle East. within 7 years. But so how did this happen? How all of a sudden you have, from no plague to 7 years of complete devastation? So you had an ancestral environmental Yersinia that evolved into a virulent ancestor, and I'll talk in a little bit some of the mechanisms by which this happened. This virulent ancestor led to what's called Yersinia pseudotuberculosis, which it's a relatively benign intestinal pathogen. Again, by benign, I mean compared to the bubonic plague. And this ended up with Yersinia pestis, and that led to what we call the Black Death, right? So how common are these pandemics? They're actually really rare. Not rare in the number of them, but rare in the agents who can cause it. Look at this. You have most of the pandemics have been caused either by Yersinia pestis, Vibrio cholerae, the agent of cholera, influenza virus, or SARS-CoV. And of course, we know, as you know, we're going through one of them. So what's this process by which a harmless organism that cannot either colonize us, or it can colonize us but not cause harm to us, emerge into a pathogen that can cause disease in humans? We call that a pathogen emergence, and this is what we call a complex phenomenon. What's a complex phenomenon? Well, it's pretty straightforward, but it's really hard to actually find an agreeable way to describe it. But I think that this can make happy most of those that are interested in complex systems. Basically, complexity characterizes the behavior of a system in which the components of the system interact in multiple ways, and leads to at least three things that I think are critical. One of them is nonlinearity. What does nonlinearity? Well, knowing what happened before doesn't give you a lot of information towards what's going to happen later. Also, collective dynamics. What are these collective dynamics? So basically, the components of the system interact in ways that are not easy to predict, and ultimately leading to emergence. Was this emergence? Well, a simple way to describe it is that the product is greater than the sum of its parts. So here you have two emergent phenomena. One of them, it's the appearance of the word, and another one that I noticed a few days ago is that there's like a typo here between the N and C, and from the outcome of a complex behavior, now you have something that sounds more like a small village in Southeast France, the emergence or something like that. So how common are these complex phenomena? Is it like just pathogens, or do we see them? And how tractable? Is it easy to understand it? Well, a good example is the economy, the stock markets. Think about it from, certainly the Dow Jones is far removed from I give you a chicken and you give me 3 fish type of economy. And clearly, how many exchanges between people have to happen for this greater emergent effect? It's really hard to define. Do we need 100,000 sales? Do we need 10,000, 1 million? Another very well known, but again, very hard to understand, complex phenomenon, it's the weather. Again, if you give me, let's say, 100 water molecules and the temperature, it's going to be pretty hard for me to tell you whether there's going to be a hurricane in Florida tomorrow or in a few weeks. Another one, and probably one of the best known and least understood ones, is consciousness. How many neurons do you need to be aware of your own existence? Is it neuron 1 billion, neuron 1 billion and 1? Of course, that's an oversimplification. And to finish up these examples, as you can tell by this, like this complex phenomenon, these emergent behaviors are everywhere. It's the emergence of novel human pathogens. Again, one thing that all of these things have in common is their intractability. because they're very hard to dissect, as you can imagine. I don't have to even go through the idea of like dissecting a brain and see how it affects consciousness. But how can you predict a couple of changes in the environment and a couple of genes moving here and there? From that to the entire world economy collapses because there's a pandemic. So one thing that we're really interested in my lab, and I'll break this slide down in the next pretty much half of the talk, it's in trying to dissect this complex phenomenon, the emergence of bacterial pathogens. So we look at it from two sides, what happens in the environment, basically what happened before they can colonize the human hosts, and also what happens once they can colonize the human hosts. We believe that by dissecting what are the molecular mechanisms and what are the ecological and environmental drivers, we might be able to not only understand these diseases better, but also develop potential therapeutic targets and surveillance networks that might allow us to predict and ideally tackle these diseases. So let me drive you through this one in our journey towards dissecting what does it take, right, to become a human pathogen. So let's start with one of the best studied mechanisms, molecular mechanisms in this case, and that allow some microorganisms to be able to harm and cause disease in humans. Excuse me. HGT, horizontal gene transfer. We can basically inherit genes in one way. It's called vertical transmission. Basically, we inherit our genes from our parents. Bacteria, besides that they can inherit genes vertically, they can also inherit genes horizontally or horizontally. There are three main mechanisms by which they can get new genetic information. One of them, it's called transformation. Basically, naked DNA gets released into the environment and a bacterium captures it, brings it inside the cell, and either integrates it in the genome or uses it out while floating in the cytosol. That gives them, like, basically it's as if you could just put a brand new app in your brain and use it. Think of it as if you just read a book, how to build wings, and all of a sudden you could actually build it. Another mechanism is conjugation, which is a way for bacteria from cell to cell to exchange genetic information. They do so using these appendages, which are typically pili, to pass on DNA from one cell to another. And finally, there's what's called transduction. Transduction, it's a mechanism that's mediated by bacteriophages. And bacteriophages are bacterial viruses that kill and harm bacteria, or at least that they can colonize them. And these viruses typically have two major life cycles. One of them is called lysogenic, where they inject their DNA in the host and stay within the genome, and as the host replicates, they replicate. And there's one called lytic. And in the lytic cycle, basically, the phage goes rogue and starts replicating without the cell, and eventually will burst the cell out and be released in the environment. As it replicates and puts its DNA within its capsid, this kind of like the head that you see, this icosahedron, it can also take some DNA from the host cell. And when it goes to a new cell and injects its DNA, not only it injects its phage DNA, but also injects the DNA of the previous host cell. So these are the main mechanisms by which bacteria can acquire new genetic information. Now, what are the vehicles in this case? Like, what are the tools, right? How do they get this kind of like, if you think of, like Mario Bros getting like different mushrooms and growing and getting more powerful. So how does a harmless commensal or an environmental bacterium get this information? So they can get them in the shape of what we call transposons, which are jumping genes, ICE elements, it's integrative conjugative elements. These are the ones that get transmitted via conjugation, these appendages. There are plasmids, which are really well known, and this is like circular DNA that encodes traits associated with virulence, such as antimicrobial resistance, toxins, so on and so forth. Similarly, bacteriophages. Again, some bacteriophages, like the one that encodes the cholera toxin, bring information into the cell and give them new traits. And those new traits, in the case of pathogens, can be used to colonize and harm the human hosts. Another really common vehicle of these virulent genes are called pathogenicity islands, or PAIs. These are like large chunk of DNA. Some of them can contain, let's say, 30 to 50 to 60 genes, and they give them, let's say, like a series of like goodies, such as a new appendage to colonize, the ability to utilize some carbon sources that other bacteria cannot, some defense systems against some predators, so on and so forth. And finally, again, these are some of the vehicles, membrane vesicles, which is basically like pieces of the cell that contains DNA within it, and neighboring cells can actually take that piece, integrate it into their membrane, and take the DNA. So as I said, these are just some of these kind of vehicles, for bacterial pathogens to gain new information so that they can emerge as a human pathogen. So here are some examples and that can explain the evolution of some of the pathogens that we've discussed. For instance, the transition from environmental Vibrio cholerae to the pandemic, not exclusively, but certainly necessitates of these steps, the acquisition of a antigen cluster, the O1. As you may recall, the disease is called by Vibrio cholerae O1. Two pathogenicity islands. Remember these large chunks of DNA that are part of this toolkit. Vibrio pathogenicity island 1 and 2. This one encodes a pilus that allows them to form like really tight colonies, what we call microcolonies. This one gives them the ability to use sialic acid, a particular amino sugar that's found in the mucus. Also the CTX5 phage, which it's a phage that encodes the cholera toxin, and as I previously mentioned, the cholera toxin, it's the source of the watery diarrhea, and the way it works, it basically creates this osmotic imbalance in the gut, in the small intestine, that leads to water leaving the epithelial cells into the lumen. And water in the lumen, it's a way of saying diarrhea. So this leads to pandemic Vibrio cholerae. Then subsequently, the bacterium continues evolving. It acquired what's called the seventh pandemic. There you go. The seventh pandemic 1, the seventh pandemic 2, a nice element. So you can see how these ingredients, when added to these environmental organisms, can give them traits that allows them to eventually be able to colonize us. Also, that gives us tools to track whether they're acquiring these quote unquote weapons to colonize us and to emerge as a pandemic threat. Not only Vibrio cholerae emerged from the acquisition of horizontally acquired elements, so did Yersinia pestis. And remember, like a few slides ago that I was talking about like these steps, right? Environmental, virulent, so the tuberculosis to pestis. So basically the environmental acquired a plasmid that gave it the ability to kind of quote unquote like handle our immune system, be able to resist phagocytosis. Then it acquired some islands that led to the emergence of Yersinia tuberculosis, several plasmids, and major genomic rearrangements mediated by insertion sequences and transposers, these jumping genes that led to Yersinia pestis. Some events of this transition are quite fascinating. One of them is allowed the bacterium to colonize the lungs. But one that I think it's particularly stunning and at the same time frightening, it's the acquisition of one simple gene that allowed the bacterium to survive in mice and be transmitted. So think about it, how it completely blew its potential away to infect humans. You had a disease that could only be transmitted from a human that gets a tick, and that tick bites another human. The minute the bacterium could survive within mouse, the transmission potential multiplied, because that tick could bite the human, take the bacterium, the tick could bite the human, take the bacterium, go into a rat, bite the rat, the bacterium could survive in the rat and spread around. That led to one of the greatest quantum leaps that you can think of in the history of humanity when it comes to pandemic, because it allowed the bacterium to spread like wildfire in the Eurasian continent. Another one is to not talk just about Yersinia and cholera, even though those two actually can cause pandemics, and that's today's topic. It's Shigella and enteroinvasive E. coli. So you have, again, these are super simplified schematics, right? You have like non-invasive, like commensal E. coli, which can live in your gut, actually lives in your gut and without harming you, and the acquisition of several pathogenicity islands, remember these large chunks, and plasmids, that circular DNA that can be acquired via transformation, led to the evolution and emergence of Shigella and enteroinvasive bacteria that can actually not only colonize, but harm the human host and cause disease in us. So one question remains here. It's something that we've been thinking about it in my lab for, I'd say, for like over a decade. It's if all it takes is like, like those video games, right, of these bacteria to swimming around or moving around, acquiring these like tools. Now I have this pathogenicity island, almost like this sort of like Pokemon cards of pathogenesis. You would think that there should be pandemics all the time, and there should be pathogens everywhere. But as you saw previously when I showed that table, there's been like pretty much 4 kind of organisms that can cause it. Remember, pestis, cholera, influenza, and SARS-CoV. So that kept us thinking, what could be the extra ingredients that you need for the assembly of a pathogen, of this emergent event to happen. And we've been very actively trying to understand that and what we call genetic variations associated with pathogen emergence. So you have like these large chunks that you acquired via horizontal gene transfer, remember transformation, phages, so on and so forth. But also we been studying like what does the core genome, what does the bacterium itself, before these mobile genetic elements kick in, need to have in order to actually emerge as a pathogen. The way I look at it, it's almost like if you give a knife to a toddler and you give a knife to a Marine, you can tell that one of them can use it in a much more different way than a toddler. So we've been looking at it from the 2 perspectives: the environment, how does the environment pre-adapt these cells, and from the host. How does the host kind of fine tune them to be an even better pathogen? So let me talk a little bit about this. So one of the things that these organisms need in order to colonize us are what we call pre-adaptations. What do we mean with pre-adaptations? Well, if you think about it, we're not so much of a training ground, if you think of it, because we have evolved to try to not give them too many chances. For instance, think about your stomach. The pH is really low, it's between 2 and 3. The reason being is that that kills pretty much almost all microorganisms. So the idea is like, we're not saying, let me give you a pH of 5 or 6 so that you can get used to, and then come back tomorrow and I'll give you a pH 3. It's like you are either ready or you're not. And that has to happen via pre-adaptation. Basically, they're ready to do this from the environment, from the environmental stage. Recently, we found that there are some genes in the core genomes of bacterial pathogens that encode what we call virulence adaptive polymorphisms. Virulence adaptive, that's pretty straightforward, and things that allow adaptations to virulence. And polymorphisms are basically changes in the DNA. For instance, in a sequence is ATCG, a change to AACG, that second A, it's a polymorphism, right? Or a SNP, single nucleotide polymorphism. So one thing we found, like for instance, pathogenic strains, and here's at the bottom, you see a picture of a porine that we study, an outer membrane porine. Porines are like, its name implies, like pores that the cells have in their outer membranes that selectively allows them to choose what comes in and out. And in many cases, gives them a lot of other properties beyond just the ability to be selective. And one of them is antimicrobial resistance. The clinical strains, in this case of Vibrio cholerae, encode a specific porine that gives them low pH resistance, resistance to bile, and resistance to antimicrobials that we produce in our intestinal mucus, whereas a lot of these environmental strains encode a completely different version, what we call alleles, that do not confer these properties. Now, what's really interesting is that this, the left version of the porine, the left allele, it's not exclusively found in clinical strains. So lots of environmentals also have that allele. So if you look at it, it's almost like if you look at all of these potential genes that can have variations, and if you have the right combination, you get like the jackpot, right? As if you were playing in the casino. It seems that the ones that can get to that next stage have the right combination of these alleles. Let's say you have in gene A, allele 1, 2, and 3, and allele 1 is the good one. So if you have in gene A, allele 1, congratulations, you can get to the next stage. And if you have in gene B, allele 3, that's the right one, congratulations. If you get the whole package, we hypothesize that that is a prerequisite for the emergence of these pathogens. So one thing that we're trying to address and test and see whether this model that I just talked about, it's correct, is to see how these genes with different here, like you have these 3 genes, the gene A is the triangle, gene B is the square, and C is the circle. The blue ones are kind of like the virulent, lame ones where they don't give you any goodies when it comes to the ability to colonize humans, and the yellow ones are the ones that actually give you these traits such as tolerance to antimicrobials. So we're looking at how dynamics in the environment of these alleles and these genes occur, and how they can be associated with the acquisition of virulence factors, with the ultimate goal is to trying to understand how can these pathogens be assembled in nature, pre-assembled in nature before they can cause disease in humans. Remember from the figure that I showed, we had 2 stages, right? We had the environment, which I just talked about, and the host. And these pathogens like evolve within us, and from a relatively benign, and in some cases, a completely benign microorganisms, they can acquire some mutations, what we call pathodaptive mutations, that can increase their pathogenic potential or give them pathogenic potential, such as the ability to colonize new tissues or the ability to produce some toxins. I'm going to give you some examples here. So in A, you have commensal E. coli, again, it colonizes your gut, no problem. And they are typically STX, Shiga toxin negative, and they are positive for one gene that's called CAD-A. Now, when these strains acquire the Shiga toxin, remember, they acquire it horizontally, but they're CAD-A positive, the toxin is inactive. CAD-A prevents the function of the Shiga toxin. However, some of them acquire mutations in this CAD-A, becoming CAD-A negative and Shiga toxin positive, and then the Shiga toxin can become active, and you have from a relatively friendly, no problem, commensal to the acquisition of some genes and point mutations in some other to have an active toxin that can cause substantial harm in the human gut. Another example, briefly, it's another gene in commensal E. coli, FIMH, that if they get a mutation, they cannot adhere into the gut, they lose that ability, but if they happen to reach the bladder, they can actually colonize and become what's called urovirulent E. coli. And similarly, this the same acquisition of point mutations in the genome applies to some lung pathogens. You have Mycobacterium abscessus, which can be a commensal, no harm. It acquires some mutations, and those mutations give it the ability, among other, to survive within phagocytes. So basically, our immune system cannot get rid of them. And something similar happens in Pseudomonas aeruginosa. It acquires some mutations, generates this mucoid type of colonies that allows them to evade the immune system and makes them resistant to what we call reactive oxygen species. Think about like peroxide, right? We use that to kill bacteria, not only when we put it on wounds, but our immune system and our cells produce that to get rid of these organisms. So we've talked so far about the molecular aspects, the acquisition. So in general, about the molecular, first about the acquisition of DNA from the environment, from other cells. Then we've discussed these more fine-tuned mutations and allelic variations, what we call these GVPEs, such as the virulence adaptive polymorphisms and the pathodaptive mutations. But what's the role of the environment in the emergence of these pathogens? Are there like some environmental drivers that foster it? As you can imagine, of course, this is not just out of the blue. They acquire some point mutations and some large chunks of genes, and voila, here's a pathogen. So here I'm going to talk about some biotic, give you one example of one biotic driver that fosters the emergence of virulence traits in bacteria. And on the abiotic end, instead of talking about pH or nutrients, I'm going to be talking about abiotic drivers that we are creating, like human-made drivers that are selecting for and fostering not only the emergence of pathogens, but also the spread of already known ones. So let's get started with the biotic ones. This is a beautiful example of what we call accidental evolution, and it's Legionella pneumophila. So Legionella, it's a disease that affects the lungs, and you can typically you acquire it from inhaling small water droplets from cooling systems. And in its natural environment, the bacterium can try to be eaten by amoeba. But of course, it has a wide array of weapons that allows it to combat these protists. And those same quote unquote tools and weapons that they use to kill and to proliferate within amoeba are used to kill and proliferate within the macrophages, the immune cells of our lungs. So you can see here like some sort of like training ground where the natural ecosystem where it lives, it has to learn some lessons to live because it's surrounded by these protists that are trying to eat them, to graze on them. And these same survival mechanisms give them traits such as toxins and effectors that allow them to colonize our lungs and survive within them. So this is like a great example of how some biotic drivers can foster the emergence of a pathogen. So how about the abiotic ones? And here I'm gonna kind of like switch slightly gears and give you a quote, because as I said, it's these man-made perturbations. And from one of my favorite movies, I think probably many of you have watched it, Princess Mononoke from Hayao Miyazaki. And really, I watched it when I was a teenager. It really kind of like made me think and want to go into the idea of, you know, how harming the environment, modifying the environment can come back and hit us back, right? And that's kind of the whole movie, how humans are damaging the forests and how the forests kicks back. And there's this sentence from Moro, the goddess, the wolf goddess, saying, typical, selfish, you think like a human. And if you haven't watched the movie, you probably have seen these little forest spirits, the Kodama, somewhere, right? And if you haven't seen it, I highly recommend. So yeah, we are harm Mother Nature, and Mother Nature doesn't take that lightly. And it's getting back at us. One of them is through global warming. What we're doing is we're increasing the water temperature. If you make the environment of waterborne pathogens cozier, they're going to proliferate, and we're seeing that already. There's strong evidence that the number of these bacteria that live in the water are increasing, and they're spreading to places where they were not able to colonize previously, because again, it's nicer for them to colonize those environments. Another example of what we're doing, the release of antibiotics. Farms typically have been using antibiotics to prevent infection, you know, in poultry and cattle, you know, so that they can mass produce these animals. And the problem is that those antibiotics go back into the environment. And what they're doing is that they're selecting for antibiotic resistant strains. So here you have like all of these little circles, the purple ones are sensitive, but there are some of them that are resistant. If you throw antibiotics, you wipe out the sensitive ones, and you give extra room for the resistant ones to proliferate and take over the population. Another way is through the indiscriminate use of fertilizers. And we see that here in Florida, where people, you know, for their, to fertilize their lawns, for orchards, for agriculture, they throw massive amounts of phosphates and nitrates. And when there's rains, those rains wash off those nutrients, and those nutrients go into ponds and lakes and estuaries. And that leads to the proliferation of like drastic changes in the ecosystem. That's not going to end up well. And in this case, it leads to the proliferation of cyanobacteria and blooms of other microorganisms that are harmful to humans. One thing that we've noticed is that actually these blooms of these cyanos, in the case of some of the ponds that we study, also lead to blooms of Vibrios, such as Vibrio cholerae and one that I'll talk about in just a second. So again, one thing that we might not see the outcome leads to a few steps forward to something that you would never think about. You add phosphates to your lawn to get a nicer grass, and you end up with a cholera outbreak. Not in this case, but if you think of the Bay of Bengal, that's not too far from actually what's happening. And finally, again, these are some examples of these abiotic changes that we are creating. Deforestation, and there are many examples of how deforestation is increasing the spread of these diseases. Like for instance, deforestation in the Amazon, it's leading to an increase of leptospira cases in the Peruvian Amazon basin, and something similar is happening in the northeast of the United States. There's changes in the forests that allow these rodents that carry this disease and these deer, these animals, to actually get in contact with humans. We're seeing more cases of Lyme disease and other spirochetes. So what can we do about all of this? I think that I gave you now the manual of how to mess things up, right? And how we are messing things up. So I'll give you with some ideas and some questions and a little bit of the work that we're doing in the lab to kind of like show you what we're trying to do to understand how this can be potentially solved, but at the very least understood. And how can we move towards a solution to this, right? Like for instance, as I mentioned, this is not a trivial problem. I hope by now, if you were not convinced at the beginning, I hope that by now you can see that this is not like a couple of little things that you can easily understand and then boom, now we know exactly what's gonna happen tomorrow. So which variables do we study? What do we look at? Do we look at changes in pH? Do we look at changes in temperature? Do we look at changes in phosphates, antibiotics, the distribution of these mobile genetic elements, amoeba? What do we look at to see exactly what's causing these bacteria to become pathogenic to humans? And not only what do we look at, but also how do we switch from a correlation, there's more of this, therefore this is causing this? Well, we have to prove that. How do you, once you choose your variable and you see some sort of correlation, end up finding a causation? You know what? Changes in these 5 variables are actually leading to this undesired outcome, because by knowing that we can either reverse it or at least prevent it or know where this might be happening. Also, if you find these variables, if you know that these variables are actually what's causing it, how often and how many samples what do you need to generate an actually accurate predictive model to actually understand how these events might happen? And finally, again, these are some questions from the dozens or hundreds that you can ask about this topic. It's like, can we now, if we understand what are the variables, what are the variables that are causing this, we have enough, well enough predictive model, can we then translate that into realistic and cost-effective surveillance framework? Think about it, and the word realistic and cost-effective matters because we cannot sample every single pond, every single piece of soil in the world all the time. So how can we find ways, to make this wicked and complex problem a tractable one? So in the final part of this talk, I'll talk a little bit about the work that we do in the lab, because I think that sometimes, we talk about things that might be a little bit general, and I wanted to also kind of show what, scientists, at least humbly, what we do on our end to try to answer some of these questions with the hope that eventually between our efforts and the efforts of some of my amazing colleagues that are doing incredible stuff, we might be able to find a potential solution to this problem. So in my lab, we have an approach that we call from base to basis, where we, punny, we collect water from the base, and then we look at we end up with molecular, we mix ecology, with bioinformatics and computational biology. We do molecular biology, and we finally study the pathogenesis, right, to understand how these organisms could cause disease in humans. So I'll give you two examples of, very briefly, one and a little bit more expanded, the second one, on how we are applying our from base to basis to understand pathogen emergence and disease. Before I do that, I want to highlight the family of bacteria that we study, because I think that once you see how diverse they are, you realize that we cannot tackle these problems by brute force. The reason being is because these organisms that we study, these waterborne pathogens, are actually natural inhabitants of this aquatic ecosystem. You cannot just bleach every pond, right? The same as you cannot survey every pond. Like one of them is Vibrio cholera, which we've been talking about. Another one is vulnificus, which causes a deadly septicemia. I'll talk about this a little bit further later on. Another organism is called Vibrio parahemolyticus that causes enteric infections, and you get it through the consumption of poorly cooked seafood and shellfish and fish. Interestingly, this is one of the other few organisms that can actually cause pandemics. The difference, and it's not in that list because this organism doesn't lead to death on the patient, and it's also self-limiting. So basically, the host will eventually clear out the bacterium, but there's one lineage that actually managed to spread throughout the world. But these bacteria not only have human pathogens in it, but also have other types of pathogens, such as Vibrio coralliticus, which it's a pathogen of corals, and it's destroying a lot of the coral reefs throughout the world, particularly associated with climate change, because the virulence genes are turned on at a certain temperature and turned off at a lower temperature. So this is very interesting and very frightening one, because it can really devastate entire coral reefs. But not only you have pathogens, you also have symbionts, such as Vibrio fischeri, which is the symbiont of the light organ of the Hawaiian bobtail squid, and also of the anglerfish. If you've seen Finding Nemo, that little organ that attracts Dory, that is a Vibrio species that actually emits light. And finally, just to give you like a broad picture of this family of aquatic bacteria, so that you can see how we cannot go heads first to tackle them. I don't know if you've heard of the puffer fish and the fugu, which is a delicacy in Japan, but it's an incredibly poisonous fish. So if you eat the wrong parts of the animal, you can die. It has an extremely potent neurotoxin. And in some species, that neurotoxin is produced by a Vibrio symbiont. So basically, the symbiont gets food from the fish and provides the fish a toxin that doesn't harm the fish, but pretty much harms everything else, making it an animal that virtually has no predators other than humans that know how to dissect it properly. So we've established some surveillance networks, and particularly we have one here in the eastern coast of Florida, where we try to take a look at how these bacterial populations evolve and change, and try to answer some questions associated with pathogen emergence. For instance, one of them that we talked about it earlier, this virulence adaptive polymorphisms, is like, do environmental strains encode these pre-adaptations to virulence? So for this, we were in a collaboration with Cheryl Whistler and Steve Jones at the University of New Hampshire. We were examining strains from the Great Bay Estuary in New Hampshire, a place that, as far as we know, has never seen a cholera outbreak. So do these environmental strains have some of these traits that could eventually make them pathogenic? Not so much A, from that potential, but B, to understand from the puzzle of the pathogen which parts actually come from the environment. And the second surveillance network that we have, one of the ones that we have. It's here in Florida, and we study Vibrio vulnificus, which I'll talk about it in a slide. And it's allowing us, the question that we're trying to understand there is like, what could be the ecological factors that select for virulence-associated traits, for these traits that help them colonize humans. So let me talk a little bit about vulnificus, which is a really interesting pathogen. It's certainly not capable of causing pandemics. So we're seeing something that basically can colonize a human and it dies with the human. How can it do that? How can it get to that first step, right, of pathogenesis before it can spread in the population and eventually, hopefully never, spread throughout the planet? So this bacterium causes a very, very nasty septicemia in susceptible hosts. It has a mortality rate paired with Ebola and the plague, about 60% if you're susceptible to it. And I'm just going to briefly show how the arm of a patient that got the disease looks like. So basically, it's what's called a flesh-eating bacterium, enough. And it really, basically spreads like within a few hours throughout your body and causes the septic shock and death. One big problem that we have with this pathogen is that it's really hard to differentiate between a strain that can cause disease in humans and a strain that cannot cause disease in humans. And you can imagine that's Houston, we have a problem. Because at least with other diseases like cholera, if you give me a strain, I can easily tell you this, don't put it in water and drink it. Whereas with vulnificus, it's a natural inhabitant of the coast. If you collect water, it's gonna be there. But most of them are not gonna cause disease, but some of them we know can cause septicemia, but we don't know exactly what makes them capable. So we know there are 3 biotypes based on some biochemistry. And we recently found that biotype 1, that's mostly pathogenic to human hosts. Let me show you again another phylogenetic tree. Remember, these trees are basically shows you the relationships of different strains, the evolutionary relationships of different strains to each other. So here we have like 4 different clusters, and we found that the biotype 1, the one that can cause septicemia in humans, they mostly fall under, actually, they all fall under this cluster 1, this C1. So now that gave us the ability to kind of, as Archimedes said, right, a point of support. Now we know which group are the ones, and we can track them. So we've been doing that for the past few years. And by looking at their genomes, we've been able to kind of look into their ecology, their lifestyle, right? And remember, cluster 1, the one on the left, it's the one that where most of the ones that can cause septicemia fall into. Remember, not all of the ones from cluster 1 can cause septicemia, but most of the ones that can cause septicemia, if not all, fall into the cluster 1. So one thing we noticed by looking at the genomes and some like phenotypes, basically behavior of the bacteria, is that the cluster 1 seems to have a very specific lifestyle, and what we call bloomer. Basically, they bloom whenever there is a sudden influx of nutrients. So we were thinking, if it has a very specific lifestyle, maybe we can find where it likes to live. Maybe it likes to live in some specific locations in the ecosystems. So we decided to take a look at places where we know that disease is endemic in the eastern coast of Florida, and started collecting water samples, sediment, oyster, cyanobacteria. Also, we were collecting small crustaceans and getting the Vibrio vulnificus from those places to see, where does it like to live? Does it like to live in the sediment? Does it like to live in associated with cyanobacteria? And to our surprise, and let me explain this figure briefly, we found that the vast majority of the strains that were from cluster 1, the ones that can cause septicemia, were coming from one location, regardless of the fraction, whether it was sediment, it was water, or it was cyanide. It doesn't matter. It was the location. So that to us was amazing, because then we can say, okay, what does this location have that fostered the proliferation of these potentially pathogenic strains? We also did some tests, and I will very briefly describe one part of this figure, because I think it's quite striking. On the left, you see the ability to survive in the presence of serum of these strains, right? So these are environmental strains that we collect from an estuary. They've not seen a human other than, you know, me and my students. And what we do is expose them to serum, which is a barrier that we produce to prevent organisms from colonizing our blood. And after a couple of hours of exposure to the serum, we see whether they were able to survive, whether they died, and one thing we didn't expect is whether they can grow on it. So if you see the red dotted line that breaks the left part in two, bacteria on the left die in the presence of serum. Bacteria right on the dotted line basically can survive in the presence of serum, and bacteria on the right end can actually thrive on it. Not only they can survive our barriers, but they can use it as a carbon source. They can grow on it. And the green ones are the C1s, the potentially pathogenic, that come from one location, and the blue ones are the C2s, basically cluster 2, the ones that don't seem pathogenic, and voila, they actually don't behave as a potentially pathogenic one, because if you die once you reach the blood, because if the serum kills you, that's the end of the journey of a pathogen. So here we have strains that are ready from the get-go to colonize, not only colonize, but proliferate within a human. So we were like, okay, so we know where they live, we know they have this potential. Is there something in that environment that might foster them? And this is where we are right now. Let me show you this final slide here. When we sample, we collect several variables. Besides collecting the bacteria themselves, we look at abiotic components, remember, pH, salinity, temperature, so on and so forth. And we also look at the community structure. Who lives there? And we are seeing some, again, we are at the early stages, right? Right now, we just have correlations. We don't have causation yet. So we're trying to look at that. But we can see some correlations between the proliferation of DC1s on the top right, the green ones, and the presence of salinity and certain species, such as Synechococcus, Cyanobium, etc. And on the other hand, we are seeing that there's a proliferation of these C2s, that C2s live in environments that have very specific sets of abiotic variables and a very specific community structure. This is kind of giving us now the ability to start kind of digging into the dissection of these environmental variables that might be fostering the selection of species or strains that have pathogenic potential. Again, step one towards becoming a human pathogen, right? So with that, I'm gonna end my talk. And I'll just, I was gonna close it with a quote from Princess Mononoke, but I don't know, I felt it was not really appropriate, because this first sentence of "you cannot change fate," I kind of like, don't think that's true anymore. I think we can change our fate. We can certainly find ways to understand these diseases, and we can find ways to eventually tackle them before they become a problem. So I decided to change it for this one that's also part of the movie, which it says, "You mustn't give up, we'll find a way. Go on planting the trees, and someday we will beat them." And with this, I would like to thank the members of my lab and the funders of my research, the NSF, the Burroughs, Wellcome, NIH, National Science Foundation, and the FDA. And I would like to thank you for hopefully listening. I don't know, I can't see anything. And here's my Twitter handle and my website if you want to know more about the research that we do in the lab. Thank you very much.
Thank you so much for the wonderful talk. And I invite people to either raise their hands down in the reactions pane, and we'll bring you up to bring a question. I see some applause reactions there. And also, people have been sending in some questions to me. They can continue to do that in the chat. So raise your hand if you want to ask one in your own voice. They're still applauding and thumbs upping you though. I'm going to start off here with a question from John, who says, I was curious if it's known from statistical analysis how many potential pandemics are out there at any given moment. For example, how many black plagues are out there at any given time, but just missing the step of the tick biting the human?
Ooh, okay. I like the end part of it, because known, that's not a problem. We know exactly, we have a cholera pandemic going on, we have the SARS-CoV-2, but that's exactly the issue. The known, right? We know that, and that's why I think that hits the nail in the head, because the one thing that we humans are really bad are the unknown unknowns, right? So we know, we know that there's a SARS-CoV-2 pandemic, and we certainly don't behave anymore as if there was one. So that is actually the critical problem, right? And that kind of like ties back, and that's why I wanted to bring that latter part of the talk on these kind of very early stages of kind of like tracking a pathogen, right? So that's what we're trying to do, track the pathogen before it becomes a problem. And most importantly, in the case of the ones that we study, to kind of understand these underlying rules of what does it take to, walk the walk towards becoming a pathogen before we get this quantum leap where it becomes out of control, like what happened with SARS-CoV-2. By the time we noticed, it was too late. It was all over the place. So yeah, great question.
Is that a matter of critical mass or something, where it just makes the quantum leap, or?
That's the problem of complex phenomena, right? We go back to that, and that's why I mentioned that. It's just it goes back to the idea of the neurons, right? How many cases do you need? How many horizontally acquired pieces of DNA do you need? How many pathodaptive mutations? Which changes do you need? There's so many variables that we don't know exactly how that leads to the recipe for disaster, which is an actual pandemic. We're trying to break it down and dissect it, but it's certainly not only it's complex, but it's complicated. So it's both ways.
Right. Okay. All right, I've got a question here from Teddy. Teddy says, you sometimes hear that it's in the interest of virulent pathogens to evolve to become less lethal so that they won't completely kill off their hosts and can continue to reproduce. Is there any truth to that?
No, that's not correct. Yeah. Yeah. It's an oversimplified way of looking at it. Again, there's so many variables like evolution doesn't have this teleological perspective, right? It's just like, for instance, with the case of Vulnificus, it just kills you and it dies with you. It doesn't get transmitted. Can it gain the ability? Yeah, it can get the ability to be even more lethal as well. And I wouldn't be surprised if it becomes capable of transmitting between hosts. So that whole idea of them becoming friendlier, are there cases of that? Yeah, but we still get diseases for a reason. So that's not truly a correct statement. Yeah, it's too broad. Yeah.
Okay, that's unfortunate.
Yeah.
Okay, Felix says, hello, plague and cholera pandemics were caused by bacteria. SARS-CoV pandemics were caused by a virus.
Yeah.
Are there any differences in the way that bacteria and virus become pathogenic? Thank you.
Great questions. So I study bacteria, but I think that there's a major, again, not major, like trend, right? You can look at it as a trend. Viruses tend to be more associated with what we call zoonotic events. So basically, a virus can colonize an animal and gains the ability to colonize us. That is typically more common in viruses. So, and that it's associated more with these pathodaptive mutations. But of course, pathadaptis. Actually, I would say pre-adaptation. Basically, you get some mutations that give you the ability to now not only colonize the lung of animal X, but also colonize the lung of animal H, human. That's kind of, again, it's a pattern because there are also bacterial zoonosis. But that would be kind of like a broad way to differentiate it. Certainly, viruses are not going to acquire, they don't have transformation, they don't have conjugation, and so forth. Yeah.
Thanks. John says, it seems to me that this kind of mixing of genes to better understand pathogenesis could also be extremely risky, e.g. the creation of Frankenstein's monster. Are there any known human-created superbugs, bacteria or virus out there? How easy would it be for a lab with basic genetic understanding to create a superbug capable of starting a pandemic?
I wouldn't do that. That's what you do is you delete them. So you take the one that can do it and take things out and see if they lose skills. We don't take one that cannot and make them get skills. So the movement of these elements is not that we give them the elements, is that they acquire the elements in the environment or in the human. That gave them the ability to colonize humans. And then now we're like with this, like in the movies, the crime scene, right? So now we have this thing that can wipe out half of humanity. How did it do it? So then we start like dissecting it, breaking down, chopping genes and getting rid of things. But we look at it from the loss of function, not from the gain of function. So we say this gene is critical because when you delete it, the bacterium cannot colonize anymore. Okay.
Hopefully people take that to heart.
No, it's not a feasible way of doing things. Yeah.
And good. Brianna asked, not positive about this, but I believe on your chart of historic plagues and their causes, the only one that had a question mark at the end was the SARS-CoV-2, COVID-19.
Yeah.
Could you discuss the possible zoonotic sources of SARS-CoV-2 versus any other possible causes? Thanks for the great talk.
Thank you. Thanks for your question. So the things like that's a table that was published, I think, like a year or two ago. It's just that I liked it because they put all of the other pandemics at once. So I think that today is pretty well established. Again, we're talking about something that's ongoing, right? That it seems to be a zoonotic event from bats. That's by now it's fairly well established. So that's a good point that you noticed that. It's just that I think the paper was published in 21, if I'm not mistaken. It's just that all the papers talking about pandemics were just focused on SARS-CoV-2, the recent ones. So yeah. That's pretty well established. Again, as scientists, we tend to never say completely fully because it's not in our nature. But pretty well means to us, I kind of done deal. Yeah.
Close to absolute as possible, I guess.
Yeah, exactly.
And reminder, you can raise your hand and ask a question if you like down in the reactions pane. I have one from Joel. It says, what are the ways that we manage COVID that work against the threat that you describe? We now have a vaccine against SARS. Have we got it in a hog tie? I guess it means solved.
I don't understand. I don't think I fully understand the question. Do you mean that have we solved the other pathogens or SARS-CoV-2? I don't know. Can you repeat the question?
Let me see. It says, it says, what are the ways that we managed COVID that work against the threat that you described? We now have a vaccine against SARS. Mm-hmm. Have we got it in a hog tie? I guess he means, is it handled?
I mean, no biological entity is handled ever. That is, it goes against the nature of nature, right? We can have it under some degree of control, but it happens with any disease, right? Particularly if we give them culture media, aka hosts, to replicate and evolve, right? That was the whole point of some mass vaccinations that eradicated some diseases. We just didn't give, by eliminating them from the population, we didn't give the viruses or the bacteria a chance to continue replicating, to continue acquiring mutations, and to continue evolving. You take away their food, but in the case of the COVID pandemic, you can look at the numbers. We're going back to our regular lives, meaning that the virus has the ability to replicate in human lungs on a daily basis. So yeah.
Okay. All right, I have a hand up here from Janu, and I'm going to ask you to unmute and ask your question.
Hey, thank you. Thank you for a timely talk on bugs and microbes. My question is about the universal vaccine, which they're trying to develop for flu and for COVID as well, I guess. What is the process and what is the difficulty in developing vaccines for like, multiple mutations? What are the challenges?
Thank you for your question. I'm first of all, disclaimer, I'm not an expert at all on vaccine development. Okay, so I just want to clarify that from the get-go. Now, I studied molecular evolution, so I can tell you from my end what could be very natural hurdles that need to be overcome. In a lot of the cases, when you develop a vaccine, you're basically telling the immune system, it's like you show the immune system the picture of somebody, right? But that picture must be substantially accurate, meaning that if the person gets like some surgery or has the eyes closed or is wearing a hat, that might make the immune system not capable of recognizing it that well. So if you take that analogy like one step further, when let's say you tell the immune system, hey, here's the spike. This is how a spike of this virus looks like. If you see this spike, go. Now, what happens if the spike changes? You see what I'm saying? Then all of a sudden, the immune system is not trained to do that. And the further it changes, the lesser the capability of the immune system to detect. Again, if you give a picture of a suspect and that suspect gets surgery, puts glasses, makeup, and a hat, then there's no way for the cameras at the airport to actually able to detect him or her. I don't know if that makes sense.
That's a very simple but a nice explanation. Thank you.
Thank you.
Okay, thank you for that. Okay, we have a question from Sarah. It says, the flesh-eating bacteria is so very, very scary. Do you ever get worried just going swimming or working with these pathogens?
That's a really good question, because we work, actually the bench that we work with is right across from my office, so I wash my hands very frequently when I work. The thing, first of all, one of the things about this disease is that I mentioned several times the word susceptible hosts. Not all hosts are created equal, meaning that if you have some underlying conditions, then the bacterium can be a major threat. That's number one. If you don't fall under that group of susceptible hosts, typically immunocompromised, in some cases people with liver problems, because, and this is a very interesting thing, the bacterium needs a lot of iron to grow. And if you have low levels of iron, aka most people, the bacterium kind of like doesn't grow at a speed that allows it to take over. However, people with liver cirrhosis or liver failure have higher iron content. So those are incredibly susceptible to the disease. But also we know how to treat it. There are, antibiotics very effectively. The big problem is that the way it works is that, think about, like, you got to look at it from the epidemiological standpoint to see also how these diseases work. In the case of vulnificus, imagine you go to the beach, you like to go fishing, or you like to go surfing, you have an open wound, and or you cut yourself and the bacterium enters it. Then you go home at 6, 7, 8 p.m. You're tired, you take a shower, have some food, and you probably got a little sunburn, which typically makes you extra tired. Then you become what we call overnights, like an incubation machine. You go to bed, and by the time you wake up, the bacterium has taken over whatever limb you got the wound. So the epidemiology really matters. You can get it also from the consumption of oysters that are contaminated with the bacterium. Again, simple epidemiology. If you eat oysters, I can almost guarantee you that you don't chew them. So they become literally like a Trojan horse, like a vehicle for the bacterium. Because remember I mentioned the low pH of the stomach, right? So now you have like them in a capsule, like literally we take medicines in capsule so that this cover protects the medicine from the acidic pH of the stomach so that it can actually reach the intestine. So that allows the bacterium to reach the small intestine. But there are some behaviors and some susceptibilities that make you kind of like likelier to get the disease. But just because it's on the bench across from my office, it's not. It's very unlikely that I can get the disease because I would have to, take a plate and kind of put it like some sort of ointment on a wound, and I avoid self killing, or adding diseases to my. So yeah, but I mean, so it's worrisome, but it can be somehow controlled.
Yeah, not to get anecdotal on you, but I have a friend whose father he was really sick and they thought he had lung cancer and all these tests and they couldn't, it turned out to be bacterial. And so they said, oh, we can treat it. And then he said, but he's weird. He had the flesh-eating bacteria too a few years ago. And
Oh, wow.
that's weird. I
Yeah.
mean, I guess he's just one of those people that is susceptible to these. He's an incubator of some sort.
Yeah, I would
I'd
recommend-
never, I thought it was deadly, only deadly. So I was shocked that he had made it through
Yeah.
that. I don't know.
Probably was not this one, because this one, you don't make it back if you really get
Yeah.
septicemia. Yeah, so that person, I would recommend that person to buy like a plastic swimming pool for the backyard and not
Never go
anywhere near like an estuary or something.
Okay.
Boring showers at home. Don't risk it. Yeah.
Yeah, it sounds like a good plan.
Yeah.
Jason has a question. I loved your quotes from the Miyazaki film. What other films or stories, perhaps sci-fi, do you think most accurately portray the threat of emerging pathogens? What lesson can we learn from fiction?
Very, very good question. Actually, I don't watch a lot of movies. Actually, I barely watch movies. But I'm really interested in novels that are associated, with disease. And if you're really interested in, the more literature, cinema, arts, and the relationship with infectious diseases, I recommend you should visit my lab's website. And at the bottom, there's talk that I gave with two comparative literature professors, which was called BioLit, Biology and Literature. And it was, the title was very clever of the organizers. It was like heartaches and stomach pains. And it was about, based on we started off from Loving the Time of Cholera from Gabriel Garcia Marquez, and how, this can be the idea of like the background of disease is a very interesting tool to develop narratives, artistic narratives. Again, another, like, I would definitely take a look at that book. A novel that I love to read, it's the Blindness by Saramago. Then The Plague by Camus. There's tons, because the thing about this is, like, think about it from the perspective of like war. Why are there so many war movies? It's the idea of disruption, right? It takes weirdness out of humanity, right? It's not like another sunny day in the neighborhood, the most boring movie you can think of, right? I go to the supermarket, buy bread, and I have my breakfast. Now, there's a deadly disease coming over, and then that takes the the strangeness out of humanity, right? Kind of the worst and the best, but it certainly creates this fertile ground for more interesting narratives. So these are some recommendations, like from the literature standpoint.
Did you ever see Contagion, that movie? Contagion?
Yeah, I watched that one. Yeah.
That's a little scary because it did start with the bat and the
Yeah, I
night
mean,
market, which was a little too close to reality.
yeah, I mean,
Yeah,
there are plenty of movies about
Yeah.
epidemics. I like the ones where they are. I like the ones that they're more on the background, because kind of like where they use it, right, as a fabric to create
The human
interesting narratives. Yeah.
Right. Okay. AC has sent in something. It says, from the New York Times, January 23rd, 2001, Australian scientists have accidentally created a virus that kills mice by crippling their immune systems, and warned that the technique may threaten to produce deadlier forms of human viruses and new kinds of biological weapons. The scientists were trying to make the mice infertile, but unintentionally created a killer. They added a gene involved in the mouse immune system to the mouse pox virus, which is a cousin of the human smallpox virus that is widely used in lab studies. Mice infected with the pathogen died, even many of those who had been vaccinated against mousepox.
Yeah.
Because people have the same immune system gene, in theory, a similar step could create a pathogen deadly to people. Okay. I missed that story.
Yeah, it's. I would have to go through the. I don't know how to put it. The permits and the politics of and the bureaucracy of working with human pathogens. But think about the fact that there's like tens of thousands of people working on these things. So the degree of safety protocols. I mean, I haven't read that, but you can see that this is the quaternary consequence of something. Like it's like these videos where you have something that hits something that hits something that falls onto something.
Yeah.
And so that a completely total unexpected consequence.
Yeah.
As you can see, it took several steps to go there. So that's going to be part of like driving. That's a car accident. But this is like, if it makes the headlines, that highlights how incredibly rare, and not only make the deadlines. The deadline required like half a paragraph to get to the point of how they ended up getting that. So I think that people overestimate the amount of time that we have. Let's think of how to create. It's not something that anybody is really thinking of. Like, let's create something for which reason. It's. I mean, I wish I could invite people to actually help me when I need to get a permit, and you'll see how non-trivial just even getting the strain of the flesh eating, to work with that one.
Yeah, I bet. Yeah. Well, this AC also asks, what about the bad actors, state-sponsored bioweapons, and the new garage biohackers?
There's.
Are they successful?
They're not successful, right? They're certainly not very successful. If they exist, they are not successful.
But it seems like if you could get that word out, what you're saying, it would kind of take away from when people were saying it was created in a lab, that COVID was created in a lab in China or something.
Yeah, I mean
that's just not even a.
I mean, there's people that think that the Earth is flat, and there's flat earthers are on the globe, right?
Yes.
So I mean, that's an extreme, right? This, I guess, it makes sense if you are not kind of like in our line of business, but it's just, I mean, you've seen how rare this is, and you've seen how many random little events must happen for a truly, thank God. I mean, look at the table. The table kind of highlighted only 4 organisms, 2 viruses and 2 bacteria that can actually cause pandemics. So thankfully, this is not a common thing to happen, which is fantastic. It's great. So it's not, and I would invite somebody that thinks that you can, on the basement, have a multi-million-dollar lab that allows you to create a pathogen, to actually visit a real lab, because it's not, again, it's not like a hacker coding a virus at home. That's not the same thing. We're talking about something like logarithmically more complex than that.
Okay. I see John said, if you're traveling at the speed of light towards Earth, the Earth is flat from your perspective.
Fair enough. You got
I
them
think-
that one, John.
John has a good answer for everything. Okay, and we have one more question from, I gotta go back there, Jason somewhere just wants to know, have we done enough to solve COVID? Have we done enough to solve COVID?
No. Yeah. I mean, do you think that we have? Yeah.
There's a lot more to go, huh?
Okay. The way I took the question is not, so I took the question from, have we taken the necessary steps in a timely fashion? Clearly no.
No.
That's the answer. If we had, I would probably be having a drink with, what's the name of the person?
Jason.
Jason.
I'm sorry, Joel. That's Joel.
Yeah, I'll be with Joel in New York right now, finishing the talk and grabbing a micronaut. But that's an example of, we are still in it. So I mean, don't get me wrong, it's easy to say in hindsight. And I'm not saying I can't believe it. Of course, there are some things that definitely are absurd. But it's when we were in the midst of it, some decisions were made that clearly were not strong and stringent enough. Some of them because there was no way for us to tell. Some of them because politics got a little bit.
Okay, I see. But scientifically, in terms of like.
Oh, scientifically, yes, yeah, but that's the
that's the easy part.
Exactly, that's the easy part. Like we like our thing. We're like, okay, let's save humanity. Let's do this now. But then there's science, and then there's reality. Now, while some people was freaking out at home, there was people working 16 hours a day. And while that was happening, there was some people at home talking about, conspiracy. So,
yeah,
there's nothing you can do. There are things you can do, but as a scientist, the job of my colleagues was, let's get something to start tackling this problem. But then these information machines, that's something that it's quite a bit beyond, because our job is not to convince the masses of things. That's and there's people whose job is just to do that.
Yeah, exactly. And who would have had the time while that was going on, while everybody was in the lab, all those hours? Well, sadly, the science is so far outpaced humanity's ability to deal with it. I don't know. That seems to be the major problem. Well, it was great having you speak with us. And Margaret has put in a link here for heartaches and stomach pains, infectious diseases in Latin American literature over in the chat. And also she has a link to your paper in there so people can scroll through. And we want to thank you, Salvador Almagro-Moreno. What a great talk. And thank you for your time and your research.
Thank you.
And everything you've done. We hope you'll come back in person.
Yay! We have the micronaut.